So this slide set that we've been going over is kind of introducing the idea of plant-plant communication, and then I wanted to cover a couple of ideas from several publications that are focusing more on the more real-world field-based studies that are messy and complicated, but there's been some really nice work done. So I wanted to talk a little bit about those. This is a chapter from a textbook, not a textbook, a book. And I posted this into Educat for reading. And so in this chapter, she's really pushing the idea of mycorrhizal networks and that you need to consider them. She's pretty much going with the idea of the holobiont theory, where you have to consider the host trees in this case and their associated mycorrhizal fungi in particular. Last week, we looked at the idea that mycorrhizal fungi can look and behave a little differently, depending on where they are, so to speak. And so you certainly have the mycorrhizal fungi that are interacting directly with the root. And with the work that Suzanne Simard is doing, she's working in forests out west in British Columbia, and a lot of the focus is on Douglas fir trees, which form ectomycorrhizal relationships with fungi. So they tend to be dominated by ectomycorrhizal fungi. And so on this, she's trying to show, the ectomycorrhizal fungi have that mantle of fungal growth over the surface of the root. So that's what's being shown here. Come on now. And so that is the mantle covering the surface of the root. And then you have to appreciate, of course, that the hyphae go out into the soil. And so you have couple of different ideas here. You have the absorptive hyphae. And then when the hyphae start to gather into cord-like structures where they kind of cluster together, all the mycelial strands cluster together, then they start thinking about rhizomorphs. That's the term that's given. And then the sclerotia are like the resting hyphae that are just hanging in the soil and can be dormant for a while. So we looked at the idea of network design in forest systems, and there's some pros and cons. Scale-free network is what she's finding in a lot of her studies and all of the people that she collaborates with. And so we talked about that last week, how you have the hubs, which in this case are the trees of different ages. And so the darker the green, the older. And the more of the black lines connecting, that's the mycorrhizal fungal connections amongst all the different trees. So you can see there's the light yellow green are the really young trees. And so we talked about this. I pulled in a table from another publication that has some of the terminology that she uses in that chapter. So you can refer to this while you're reading that chapter. And yeah, so she has in there the hub, links, and then she's also talking about different network designs. Okay. When they've been looking at these interactions between plants, we talked about the different things that can get transferred. We'll get into this a little bit more, but I mean, it makes sense. They've known about carbon transfer and nitrogen and phosphorus and water for a while now, but some of these other ideas have been proposed as well. So they're tracking all of this. They're trying to see how this information flows and what part the mycorrhizal fungi might play in this in real field sites, real systems. Glutamate and glycine are interesting because they're amino acids that are amino acids have nitrogen. And so that is kind of the preferred form of transferring nitrogen-based nutrients And they're actually neurotransmitters that we've been talking about way back. And when we looked at plant behavior and neurobiology, they're finding that glutamate especially seems to behave similar to how glutamate works in nerve systems in humans and animals. We'll get into sound a little bit. We'll see what's up with that. And then we, are acknowledging that all of the chemicals and communication signals that are getting transferred, the hypothesis is that they're going to be influencing growth and survival. And the idea that the mycorrhizal fungi are there in such an abundant amount through the forest system, it really does point to the idea that they can be like the holobiont idea, where the microorganisms that coexist with the host, they are very capable of adapting on the fly compared to the host tree, for example, that is very long-lived, a little slower to react, and isn't nearly as able to adjust so quickly like the mycorrhizal fungi can. So you have to imagine in these forested systems that you have all these mycorrhizal hyphal growth forms, and the idea that you have multiple trees and even amongst different species, and not just the trees, there's also the shrubs and understory growth too to think about, that they're all potentially connected, interconnected with the mycorrhizal fungi. And of course, the mycorrhizal fungi has intimate connections with the plant roots and allowing for transfer of water and minerals. So just refreshing your memory to think about the mantle covering the root surface, if it's an ectomycorrhizal, all the hyphae going out into the soil. So just getting that back up in your head of how they're forming, and I'm kind of imagining that over the forest system, and that once the plant is taking up, the roots are taking up these water and minerals and other chemicals, they could easily get put into the root tissue, into the vascular tissue, and then through the whole plant body. And so it's a very intimate way of transferring nutrients from the fungus to the trees. Make sure to go over, is it this one? No, where is it? We talked about in this chapter, she talks about crystallized versus fluid intelligence and gets into how the mycorrhizal network display both of those. And so she says, they can see that there's some networks that have weak links that are growing very rapidly, and they're very dynamic and they can change very rapidly. And so she's making the analogy that's similar to the learning process in animal and human brains. So that's like the fluid intelligence. And then with the rhizomorphs, the ones that have the clusters of mycelial hyphae growing together, she says those are stronger links, and they tend to be able to go very long distances in the underground in the soil system without damage, and are more akin to the idea of crystallized intelligence. So she'll make these comparisons in that chapter as well. Okay. We saw this too, how, she's saying, okay, the root brain idea that the Darwins proposed, let's take it and a step further now that we know how important mycorrhizal fungi are, and extend it to the networks of mycorrhizal fungi as well. And, because they're so quick to grow and they're flexible and adaptive to their environment, much more flexible than the longer lived tree that's slower to respond. And so, I mean, that makes sense considering when we evolved, when trees evolved, plants evolved, the earth was swimming in microbes. I mean, microbes were everywhere. You can't get away from them. And so it makes sense that there are these dynamic interactions between, in this case, the host plant and the associated microbes. So in that chapter, she's looking at this idea of, with the short distance hyphal growth in the underground, ECM is ectomycorrhizal fungi. Those hyphae tend to be very active, they're dynamic, so they're growing, and if an area that they grow into isn't very useful, they'll prune away that part and grow in another way. And so it's very dynamic in terms of their foraging and kind of adjusting their growth and behavior depending on the patchiness of the soil and what's happening. And she makes the analogy that's kind of what our brain's doing when you're going through the learning process. You do weed out things that you don't use anymore and lay down new networks for things that you do. And then with the challenge of the mycorrhizal fungi growing through longer sections of the soil system, she is making the analogy of the rhizomorphs with their tougher, stronger links, um, but that they are very good at transferring nutrients to the trees they're associated with. And it's more like the crystallized intelligence. When you finally do learn something and it's in your long-term memory, you have a strengthened neural network with many more neuronal linkages. And so, kind of an interesting analogy there, kind of taking Charles Darwin and his sons Francis's ideas and broadening it out into the mycorrhizal network. So with her work, she has been focused on forests that are dominated by Douglas fir. Douglas fir is a economically important tree. It is harvested regularly. And one of the studies that she did was in a Douglas fir stand that had different ages of Douglas fir trees in the stand. In this particular case, it was this study, there are many ectomycorrhizal fungi that can hang out in the different forest systems. They had the ability to focus on two ectomycorrhizal fungi. So there were many more there, and they acknowledged that, but financially, you can't cover them all. So they picked Rhizopogon, which is incredibly dominant in Douglas fir systems. And so they were looking at two different Rhizopogon ectomycorrhizal fungi. They display both that short distance hyphal growth as well as the rhizomorph growth. And they can point to the idea that all the rhizopogon, they are connecting these trees in a network underground, and that they're continuous complex networks. So you might have heard the phrase the wood wide web. She was the one that coined that, the wood wide web. And that kind of made the headlines when this study first came out because it was a big deal. No one had really done research on a big field scale like this to really demonstrate that you have all these mycorrhizal fungi underground connecting these different plants in the forested system. And so when she was doing all this work initially, she did show that these forested systems with these uneven aged Douglas fir do that scale-free idea, which is back to this idea where you have the idea of with scale-free, where is it in here? Also note that, where is it? No. So random networks are more resilient to perturbations. So that's this idea. But she's saying the forested systems there in British Columbia are scale free. So they're more susceptible to perturbation and slower to recovery potentially. And you have these links where you have major hubs and then you have them connected to many other trees in the area. And so this work, again, going back, this is some of her data with some of her grad students and postdocs, where she's showing the different hub trees in the mycorrhizal network. So scale free connections. Whoops, wrong one. Okay. And so those large hub trees were linked to each other, so hub to hub, but then they were also linked to the younger trees too. but the younger trees had fewer connections between other younger trees. So it was more of a um, older trees to younger trees was a stronger connection. And so when they were doing some of these early studies, going back to this picture again, the one with the arrow there on that, that was like the mother tree. They referred to it because that was the one with the strongest connections. For that one was linked to 47 other trees and was estimated to be linked to at least 250 more trees if the larger stand had been sampled. So they only did a 30 meter by 30 meter patch, and they were getting this kind of data. And so this quote was taken from that paper in the conclusion section, to ensure that old growth Douglas fir forests remain resilient and self-regenerative following disturbance. Our findings support a management approach that conserves large trees or groups of trees and their mycorrhizal fungal associates. So what's the preferred way of, well, what's the historically preferred way of logging? Are you familiar? What do they normally do when they go to log trees? Clear cut, right? Clear cut. And they often use machinery that can be very damaging to whatever is in its path, not just the trees. So we'll come back to that idea because I'm going to talk a little bit more about some further work that she's been doing on this idea of how do we maintain. So she's actually works in a forestry department in at the university at British Columbia. So she's a trained forester. But what I really like about her, I guess her angle of research is that she's looking at, are there better ways to do forest management where we can still make a profit, but have the forests in a safer, healthier system through time. And so I'll come back to this idea. So this common mycorrhizal network with the Wood Wide Web that her group established in some very careful research was met with a lot of criticism. And so there were several scientists that criticized that, no, this really can't be happening to that extent. You did it wrong. There's some bias. And so there was some interesting, especially it was in 2023 where they really started picking on her, I guess. Even though she published this work a long time ago, what I just talked about, that work, that has been done quite a bit ago. And it's not just her, there's been other scientists at other universities around the world that have demonstrated similar findings. But in 2023, there was a group, it's interesting, Karst, the last name is Karst in 2023, Henriksen and Robinson, all the year 2023, started to criticize. And Simard says it happened, funnily enough, after she published her book, which is Finding the Mother Tree. That's the book that she wrote. It's kind of like an autobiography slash I mean, she talks a lot about her research, but she also talks about her perspectives and, about how she got into her current, training and research. And after that was published and met with some, good success, then there's these criticisms. And so this, I'm not going to post it, you can find it if you want, but this opinion paper that's written by her and a couple of other collaborators, it says, response to questions about common mycorrhizal networks. So those three papers in 2023 were saying that no, common mycorrhizal networks to that extent just really aren't happening. I found it interesting because I'm like, Karst, where do I recognize that name? Karst. And then I started looking. So she's the one that published this top paper. And you can see, she's saying citation bias. And then the other papers too were, criticizing. And yet these papers down here was Karst and Simard working together. So 2006 was the earliest paper I could find where Karst is in there. is Sylvan Samar. And then this paper, she's first author, which is a big deal. That means she did most of the work. And Suzanne Samard is there. And this one is to 2020. And they're both on that paper. So what the heck happened? There's some kind of drama going on there. And I don't know. I don't know them personally, but they both, are on multiple papers together. And yet all of a sudden there's this big argument. So I don't know. Take that as you will, but it's science is done by real people with opinions and egos and biases. So, stuff happens. Okay, so this is another paper, a famous paper. Brian Pickles worked in Suzanne Simard's lab, and this was one of the more careful studies. So when they met with some criticism about this idea that things are getting transferred via the common mycorrhizal network, they performed this study. So this one was more of a lab-based study because it's easier to control all the variables, and they wanted to really nail down what was going on because of some of the pushback they were getting. So this is in 2017 when this was published. And so they're saying, yep, we can show that these networks do transfer stuff like water and other things. And so they focused on carbon and they worked with Douglas fir and they were looking at kin behavior too. They were looking at that. So they were looking at a lot of things in this paper. But the second bullet point was referring to the kin, closely related plants display greater network size. And so they know that that's been published by other scientists that work with arbuscular mycorrhizae. They're like, yep, they're showing that they have this kin idea. And so they're asking, could this happen in Douglas fir with ectomycorrhizal fungi? And so they decided to explore this idea. And so a lot of work is done with the arbuscular mycorrhizal fungi because a lot of those plants are plants we care about for crop production. But she wanted to look to see with ectomycorrhizal fungi what would happen. So they did some greenhouse experiments. And what they did is they had different ages and sizes of Douglas fir seedlings. So they were 11 months old and 3 months old when they were taking the data. They had them growing in the same pot, and then they were separated by a mesh barrier, two different kinds. Some of the mesh barriers had larger pore diameters, pore mesh sizes, and another one had smaller. And then they exposed the older tree to radioactive carbon. And of course, that gets turned into sugar that can then potentially get transferred somewhere. And so they were looking at that idea. So those different meshes were basically to make sure roots couldn't touch between the older and the younger, or they refer to them as donor and recipient. So the larger mesh, 35 micrometer, should make it so that the roots can't get through. Or the fungi, the ectomycorrhizal fungi at 0.5, they shouldn't be able to get through 0.5 micrometer mesh size. So in one pot, they paired up these Douglas fir seedlings. And they were looking at where does the carbon go? If they give radioactive carbon to the older tree, what happens? And they were looking at what's going on with the mycorrhizal fungi, but they were also looking at what's happening in the soil too. So they really did a pretty complicated experimental design that was thinking of a lot of different aspects to test. Okay, so let's look at what they were doing here. So this is a mock-up of how they set up the experiment. So the D is donor, that's the older tree, and the R is recipient, that's the younger tree. Then you can see that there's this mesh by the dotted lines there. Those are the mesh bags. So the mesh bags are separating the two seedlings. So they were looking at in figure A here on this one, they were testing the above ground, right? A is above ground. There I got the right one. Yeah, one donor and one full sibling. Oh, they're just showing the basic design. And then B is where they were testing the above ground biomass. So the A is just like, this is in general what we did. B, what they did at the end of the experiment was they tested the above ground biomass, so the shoot and leaves. They also tested the below ground, so root mass. So it's being shown in figure C. Figure D, they tested the ectomycorrhizal fungi, the root tips that were covered in the mantle, because you can visually see that. It's easy to do. So they did a comparison between donor and recipient of the mycorrhizal fungi. And then E, that one, they said rhizoplane. So what they did was they removed the seedlings from the pots and just gently shook the root system, and whatever soil was still hanging on, they called that the rhizoplane soil. And then the last container there in figure F, that's where they took the kind of like the bulk soil that was left in the pots after they got rid of the seedlings. So lots of samples, very carefully taken, and following the radioactive carbon. So let's look at some of those results. So in the kin versus non-kin idea, when they were looking at so they knew the seed sources that they planted, they knew if they were kin or not. And you can see here very different appearances to those regression lines. So here they're saying the mycorrhizal abundance in the donor and recipient. You see this nice positive correlation if they were kin. Non-kin, there's no significance. So the p-value is way above 0.05. So it's definitely not significant. You can see it's much more flat. Data's all over the place. Whereas the kin, you can see it was definitely significant. So this idea of the mycorrhizal fungi abundance, definitely positive relationship with the kin idea. Okay, so that was something that was kind of peripheral to some of their work. They did note that, but they were really interested in what happens to that carbon. And so this is some of the results from that paper where so the D stands for donor on this graph, donor root, donor mycorrhizal fungi, the rhizoplane, rhizosphere, and then just the remaining soil in the pot, and then the mesh. In the pot there, the mesh that separated donor from recipient. And they were tracking where does the carbon go. And so that this is the look at the different groups of microbes essentially and the plants, like where does it end up? And so here you can see, of course, the root is plants, but the mycorrhizal fungi here, that's a plant fungus interaction, and then the interface in the soil, and then all the way to the plant fungus in the receiver. Does that make sense? So they're kind of tracking what's going on in the flow of carbon. And so you can see that they were looking at, yes, the plant and the mycorrhizal fungi. So the purple, that's the mycorrhizal fungi. So you can see that a lot of the carbon was ending up in the fungi, even over into the recipient. Okay, so it made it over into the recipient plant and the recipient roots. But they were also looking at where else does it go? And certainly the donor mycorrhizal fungi had some of that carbon, which makes sense. It's feeding its symbiont. But you can see that some of it, so to speak, was leaked out, where you can have the gram-negative and gram-positive bacteria that are hanging out near the roots, also gaining a little bit of that, which we've talked about already in this class, how the microbes near the roots in the rhizosphere benefit because they get things that leak out from the roots. And so this was a really nice study showing that to be the case with this radio labeled sugars, essentially. And they could show the flow from the plant to the mycorrhizal fungi to some of the surrounding microbes, but also through the mesh where there's no physical contact and still getting up into the plant. By the way, they did this through phospholipid phospholipid fatty acid profiles. So I've talked about that in microbial ecology. It's a pretty slick procedure where you can take a soil sample and essentially extract out the phospholipid fatty acids from all the living organisms there, and you can tell who's there just based on that. It's really kind of a slick procedure. Okay. So the data did show that the fungi really did get the lion's share of the carbon, but they are concluding that the fungi were also playing a role in getting it transferred through to the receiver, to the younger Douglas fir seedling. So when they were closely related and they were hanging out in the same pot together, they had better carbon transfer over to the receiver. And it was quite notable. So they're saying, yeah, it does seem that if they, are in the same pot and they're all kin, it does seem to influence how the carbon transfer occurs between the donor and the recipient. And so she's showing, I think this is one of the first papers that could show it in an ectomycorrhizal, a conifer setting rather than just the more typical plants that are associated with the VAM fungi or the arbuscular fungi. And so this was kind of the first attempt at that in a conifer type setting, and saying that we really do need to think about this. And of course, she's then taking it to think about what happens in a more realistic scale in some of these forested systems. So this lab-based study was very carefully done and helped kind of support what she was finding in the field because when she published those field studies, she was getting some pushback. So they went into the more controlled setting of the greenhouse. So then this study, which was actually one that has Karst as the first author, Suzanne Simard's group was looking at how do the mycorrhizal fungi impact this bark beetle outbreak that was going on in the Northwest. And so again, looking at bark beetle. So this is the scientific name of the bark beetle. And Pinus contorta is lodgepole pine. The lodgepole pine trees are the favorite host, I guess, of the mountain pine beetle is the name of the common name. And they were looking at this idea of, okay, what happens when you have an infestation? And so they were looking at, can this can the experience that the plants have, can that get transgenerationally passed on into the pine seedlings into the next generation? So they were looking at that. So we've talked a little bit about epigenetics already in the transgenerational. And so they were looking at what happens if we follow this out into the next generation in stands that were killed by the beetles or in a control stand that was left, no beetles. So that was their control. And so they looked at many different variables that they were testing, but they did show that the fungal communities did differ. And so they were collecting, what they did was they grew seedlings from those plants that had, they collected the seeds from the plants that were being attacked or in the control stands, and they grew them up in fungi, in soil that had fungi from the infested stands or in the uninfested stands, the control stands. And they were showing that there was lower monoterpene concentrations. So monoterpenes are terpenoids that get produced in response to attack, typically by insects. And so it was kind of weird. They're like, it had lower terpene concentrations compared to seedlings that they grew from undisturbed stands of, so the control stands. So they were showing that the stand mortality caused by prior beetle attacks have cascading effects on the next generation, the seedlings that grew after that initial attack. Their chemistry shifted, and as well as their growth and survival. But their view of it was they were specifically interested in what role do the mycorrhizal fungi play in this transgenerational memory idea going into the next generation. And so they were really focusing in on what happens in the the beetle attacked system versus the control. So this is a big deal. This map is showing the range of this beetle, and it does attack other species besides lodgepole pine, but a lot of the studies are done on that one because it's an important economic tree species. But British Columbia, you can see it's just a whole big swath in that area of Canada and then down into the United States. This is visually what it looks like when you fly over a stand that's being decimated. So they call it MPB for short, mountain pine beetle. And so they're native to the Canada and North America area. It's not like they've been brought over somehow. They're native. And they really like lodgepole and ponderosa pine, limber pine, and all the way into ornamental pines when the disease gets really bad. And if you are ever out west and you see anything like this is good evidence. Those little pitch tubes, they call them, that are kind of oozing out on the tree trunk and kind of look like popcorn kernels. So that's what you look for. And the beetle has 1 year to do its life cycle on this. It has an egg stage, larva, pupa, and adult. And so the female sets up camp. She goes and finds a tree. And then once she's found a little home for herself in that tree trunk, she'll put out a pheromone. So then the male comes over and then they mate and then she deposits the eggs. I'll show you what they mean by the gallery. They actually call it an egg gallery. It's underneath the bark. And then the eggs hatch in about 1 to 2 weeks, and then the larvae start to tunnel away from the initial egg gallery. And then they stay under there until the spring, and then they pupate and come out in the spring. And then the adult finally emerges and goes and finds another tree and does it all over again. Very successful from the beetles' perspective. They have been very successful at creating a life for themselves doing this method. So this is just a visual of what we just described. And you can see the galleries here where you have the like the central aisle and then the side sheets off of it. And so the female is busy, creating a little home, and then deposits her eggs, and then the maturation process through the rest of the life stages happens there. So in the process of this, it is damaging the tree. So we have to remember now in the bark area, that's where the phloem, the active phloem is located. That's the part of the vascular tissue that does all the sugar movement through the plant body. So if you start disrupting phloem movement because of all this activity and ingress, that's really bad for the tree. The tree essentially starves to death slowly. So that's an actual beetle gallery that they've took after they pried off the bark in that section. And then when the adult comes out, you'll see these holes, very almost perfect circular holes as they bore their way out and start to fly to the next tree and do it all over again. So very, very damaging. The other thing that they do, the beetle tends to have a pathogenic fungus that comes with it. I'll mention that again because that comes up in another paper that I want to talk to you about. And so not only is it the beetle doing the damage to the phloem and causing issues with that, but often they carry this pathogenic fungus that also attacks the tree. And so can be a double whammy if that fungus is coming along with the beetle. Okay, so in their work, when they were looking at this, the mountain pine beetle definitely was changing what kinds of mycorrhizal fungi were showing up. So if you look at that top bar graph, and you can see the legend there is describing all the different parts of the bar graph are different mycorrhizal fungi. So you can see just by an initial look at it that those two bar graphs look different from each other. You have in the beetle killed, you can see this huge shift here with that's just one example, with one of the mycorrhizal fungi. And so, just taken overall, both those bar graphs, very different from each other. So definitely that's supporting the idea that the infestation by those beetles is changing the mycorrhizal fungi that are there in that plot. They were showing that the mycorrhizal fungi were also influencing how much monoterpenes were getting produced by the plant, by the seedlings that were being grown. The seedling survival was really bad in the field plots that had the beetle infestation prior to their experiment, which kind of makes sense. So there's this holdover legacy. So they were saying, maybe when you think about trees that live so long and their timeline is just slowed down compared to us overall, that maybe they have to rely on the mycorrhizal fungi and other symbiotic organisms as a way to try to deal with these infestations that can hit them so quickly and so frequently. But they're saying that, taken together with this data that they were collecting, they're showing that there is this transgenerational impact because their seedlings definitely were impacted, as well as the mycorrhizal fungi in those plots. So it extends far beyond this single cohort of trees that were initially attacked. Transgenerational cascading effects of this native invader can influence subsequent generations through disrupted below ground mutualisms. So that last phrase, really important, because when you look at literature where they're looking at this type of idea, like how do you replant an area that's been wiped out due to beetle infestation or knapweed, infestation in an area. How do you deal with this? And they've often had issues with, they go into these fields that were decimated by something, a beetle in this case, and when they try to replant, it doesn't work so well. And no one until fairly recently thought to ask, maybe it's because something's changed underground. And now the trees that were there, you don't have the mycorrhizal fungi there either. And so when you try to replant those species, those trees, there's nothing there for them to work with in terms of their symbiotic partners. And there's been many, many cases of failed replantings, and it doesn't have to do just with insect disease. Like the spotted knapweed idea is also an issue, where I know of several students in the summer, that's their summer job. They go and pull spotted knapweed from areas, and it's a non-native plant. It looks real pretty, has those purple flowers, and people, if they see a big field as you're driving down the highway, like, oh, look at that really cool plant. It's like, no, it's evil, spotted knapweed. So, but you pull that and then you go back in and try to replant it to all the native plants that were there, and often it doesn't work. And it's because they now know the spotted knapweed have changed the underground composition of the mycorrhizal fungi in particular, but also the bacteria there. There's been some nice studies showing that. So this was being shown in this case with mountain pine beetle, that you do have that shift and it can influence the next generation not being able to get established. And so this was, well, this in this graph here on the bottom is looking at terpene levels. And so again, you can see major differences. So somehow they're saying the mycorrhizal fungi, because they were so decimated in the beetle plot, that they wonder if they were playing, it seems like they were playing a major role. If you can see in the undisturbed site, they still have good amount of terpene production. They have healthy mycorrhizal fungi. So they're pointing to, we got to think about what's happening underground, in that the ability of the plant to defend itself, in this case, it may be heavily influenced by the mycorrhizal fungi. And now that they're not there or are shifted in their species composition, when you plant those seedlings into that previously infested site, they're not going to do so well. So it was a really, really nice study looking at this particular insect disease that's starting to, well, it's been a problem for quite some time now, and it's not going away. It doesn't seem like that beetle is disappearing. Okay. Oh, we got to take a break. Why don't we take a break? What time is it? It's 2:52, so 3:02-ish. We'll start up again. All right. Okay, so Samard's group was collecting some good data showing that the presence of the beetle infestation was causing effects into the next generation of seedlings, Douglas fir seedlings, and she was focused in on underground stuff, like what's going on underground. So her data was showing that because the mycorrhizal composition has been altered in the beetle-infested stand, it impacted the seedlings that were grown there into the next generation. And that, you have to think about the underground in terms of what's going to happen into succeeding generations, and that you can't ignore that there could have been this shift in the mycorrhizal network in particular and the impacts of that. So then another group of scientists a couple of years later Still looking at the mountain pine beetle, they said, could we do a field study where we look at volatiles? So now they're above ground. They're like, what about volatiles that an attacked tree might give off? Can we see if that does anything to neighboring trees? And so they looked at the effect of volatile compounds. They did an actual controlled study. So unfortunately, they had to infect a tree for this. And what they did to serve as a proxy, they used the fungus, not the actual beetle, which at least I'm grateful they did that they only worked with the fungus. So this is the fungus I was telling you about that can come with the beetle and cause problems for the tree. So they just worked with that. And I'll show you that picture again. That's kind of their graphic of what they did in their research design. So we'll talk a little bit about that. But the fungus they chose was the blue stain fungus. Have any of you seen that here? It's over, I see it quite a bit actually. Actually, I like the look of how it looks. I actually collect some of those branches and tree bits because they can look quite dramatic. But this blue stain fungus, Grossmannia, associates with the beetle. And the beetle will carry the spores of the fungus or some of the mycelia, and they have it on their bodies. And then they have these special structures, the beetle does on their head that are called mesangia. It's just like a tissue area that's reserved for its gatherings of the spores. And as the beetle is digging around under the bark, the fungus is deposited and starts to mess with the flow again of water and sugar in the plant. And so it can cause, along with the beetle, it's kind of a 1-2 punch for the tree. Now, they did a field study, and I give them credit, that cannot have been easy. So doing a field study where you're trying to assess VOC dispersal, pretty amazing that they were able to get some data on this. I'm not going to get into all of the design that they did or all of the results, but basically they're saying we would like to try to do this in a complex system, not in nice, tidy little growth chambers. So let's go with that. Let's do complex. So of course they had to think about the slope and aspect of the land and the plot of trees that it was situated on, how many plants were there altogether. It's certainly going to influence the VOC distribution. All of the tree attributes, they had to, deal with all of that. So they had a lot of data to collect in this field plot. They had to look at all the trees that they were working with in terms of age, different species, sizes. And then, of course, you can't ignore the wind. They had to keep track of wind on a minute by minute basis as they were doing the data analysis. And so lots to think about. They must have had an army of grad students and undergrads working on this because that could not have been easy. Okay. So this graphic is trying to show that in the center is where they put down the fungus. So this darkest tree here, right in the center, that's the poor tree that got the fungus. And they're trying to, you can see some of the chemical structures that they isolated. So they were isolating the VOCs, of course, from the plant that they infested with the fungus. And then they would assess the volatiles given off by nearby trees and then, out into the distance. So it kind of makes sense that what their data was showing, the volatile compounds were evident after they put the blue stain fungus in there. It depended on how far away they were getting from the actual tree they infected and the site aspects. I mean, that kind of makes sense, depends on the wind direction, how far it travels. But one of the very interesting findings was that they could show that it was really only very closely related trees in terms of their chemotypes. So this is looking at type of secondary metabolites that they make. And if you have similar secondary metabolite production, you're chemotypically similar. And so it's like a proxy for kin versus non-kin. You can think of it that way. And so they were looking at studying the different volatile compounds that were being given off by all these different trees after they started the experiment. And they're showing that the plant that was infected seemed to, you would get the chemotypes popping up in related and not the strangers. So in a kin compared to the strangers. So these results suggest that plants discriminate between chemical cues from the kin and strangers. And the emitters likely aid only in chemotypically related plants. It was like a private conversation where the plant they infested, they studied its chemotype and showed that only similar chemotype plants were getting the signal to ramp up. And so uh, amazing that they were able to get some data to draw some pretty sound conclusions that you have this kin conversation, kind of like a private conversation going on in this mixed forest system. And so this was one of the first published where they were dealing with a messier system like this instead of looking at a greenhouse or growth chamber studies. And so we'll talk more about kin versus non-kin. That's kind of the second topic we're going to get into after these forest studies that we're looking at. But this idea of volatiles, of course, we're going to come back to this too, because this is as we talked a while ago when we looked at volatile communication, it's a pretty easy way for trees or plants in general to send out signals. It's pretty quick. And in this case, they were showing it seems like it was a very private conversation. The attacked tree was only sending out signals to chemotypically similar trees nearby. But I wanted to talk about this too. This is another forested study done by Samard's group where this was done in 2021, so fairly recent. This is really, I was so happy to see that this was being looked at, where again, they're looking at if you want to do logging, logging is never going to stop. We're always going to be logging trees. So if you're going to do that, is there smarter ways to do it? And so she's looking at, instead of clear cutting, could we do something else and have then the next generation of trees grow that much better and healthier and quicker, which would only support the forestry industry. So she's like, instead of being greedy now and clear cutting everything, can we kind of temper that? Is there a better way to do it where you still end up getting a payout in terms of economic prosperity from these efforts? So she was looking, her group, as you can see, a lot of people involved in this effort. And this is up in British Columbia again, in Vancouver. And so lots of people involved. So in this is kind of like the abstract. So here she's saying, clear cutting, when you just clear the whole site of everything, all the trees, everything gone, and then of course you're bulldozing over all the shrubs and smaller plants too as you're doing it. You reduce the proximity to seed sources for the next generation and the mycorrhizal inoculum potential for any seedlings that are going to regenerate in that stand. Partial retention of legacy trees and protection of refuge plants. So she's saying refuge plants with regard to mycorrhizal fungi. As well as preservation of the forest floor. So the leaf litter and all of that and she also brings up mosses that are on the forest floor. They can maintain mycorrhizal networks and help colonize the new seedlings. The germinants are the new seeds that germinate and grow. But she's saying, even though this is the year 2021 when they published, we just don't know that much about what's the best way to do this so that we get enough of the mycorrhizal fungi persisting, we get good seedling germination after the cutting has happened is there a way to do this besides clear cutting? So they did an experiment. I'll come back to this. They did an experiment where they use this kind of machinery. I think it's called a feller cutter, cutter feller, something like that. I'm not a forester, but you've probably seen those in videos at some point where they just go and they grasp the tree trunk and then cut it. And so it's a biggish piece of equipment going into a stand. It's on those track wheels, so lots of damage. So the experimental design that they were doing, they had one field, well, they had several, they did this in replicates of 4. So they had fields that were clear cut. That means no plants were retained. Then they had what they called the seed tree plot, which is they kept 25 large Douglas fir stems per hectare at 25-meter spacing. So about a 10% dispersed retention. So 10%, let's just 10% retention of those trees. And then they did a 30% patch retention, 60%, and then a control where they left it undisturbed, 100% retention. So that was their experimental design. And then what they did was they had the cutting take place, and then they waited 3 years for regeneration. So long-term study, kind of have some patience, and then went in and started collecting data on what has regenerated over those 3 years in these different stands. So let me go back. I'll go to this one. So Douglas fir, when they produce the seeds in their cones, they're wind dispersed. Okay, so that's important to think about. It's wind dispersal. And here they're showing that if you have the so-called seed trees, so some of those Douglas firs that were retained in those different percent patches. So you can see it's 0 to 5 meters. You can see that there's a lot of seedling density, but you can see that there's this general trend as you go distance from those Douglas fir trees. And so this data does make sense considering it's wind dispersal, that you're going to get clusters closer to where the mother tree was. But they made the point of saying that even out to beyond 60 meters, they said even this kind of regeneration is adequate for economic, like it's good, from the forestry practices perspective, that even beyond that is good. So they're saying that, they were able to show that if you retain some of the Douglas fir, you are still going to end up with trees. But the data does make sense. They're wind dispersed. This is looking at what's going on to the other plants besides the trees. So when you bring in those bigger pieces of equipment to do the clear cutting, you're going to be also bumping into and tracing over the bryodes. That's the mosses, primarily mosses into that category. And you have herb shrubs and then the black trees. And so the clear cut, you can see the pre-clear cut, there's some pretty good richness. So you're looking at just, the number of different plants here. And then of course, post, you can see a drastic decline in the clear cut. And then if you compare pre-post on all the others, you don't see that drastic of a hit compared to clear cut. And so even a 10% retention got you some better results. And 30% and 60%, you can see especially the 60% is kind of close to the control. What you see there, that's pretty kind of good. Well, even 30%. And it's almost, look at how it's reversed. 30% pre-post is reversed compared to pre-post here. Kind of interesting. So the point they're trying to make here with this data is that when you're looking at regenerating forests for forestry production, you can't ignore these other species in terms of the impact they have on seedling germination, mycorrhizal fungi, ability to persist, the moisture, soil coverage, all of that is coming into play and allowing for, they generally say the more diverse a forest is, the more healthy it is. And that, of course, diversity in different plants. So this data was very nice to show that you can get some nice richness of these species with a more conservative cut rather than the clear cut. Questions on that one? Some other data. So here they're looking at different types of mycorrhizal fungi. So again, what happens when you do these different forestry cuttings? What's going to happen to the populations of the mycorrhizal fungi? And so with Douglas fir, they prefer to hang out with ectomycorrhizal fungi. And so they were keying in, definitely going to test that. And so the green box is showing, green part of the bar is showing ectomycorrhizal. So again, you can see that with a clear cut, and this time they took data at 1 year and 3 years for the fungi. They were able, look at how the ectomycorrhizal got tanked in the first year. There's hardly anything there. And then it did bounce back a little bit. But then you start comparing the clear cut to some of these other cutting where they did the 10%, 30, 60, and you can see much better recovery of the ectomycorrhizal fungi. So again, they're showing that when you do a clear cut, it's not conducive to maintaining good mycorrhizal fungi populations. And often when you have that much sunlight on the forest floor, that's not used to that, the mycorrhizal fungi aren't going to do well on that either, especially when you think about the fruiting bodies and spores and all of that, it's going to be impacting. And you can see with the 10%, 30%, and 60% that you have better recovery. And then they were also tracking. The one thing that can happen sometimes in some of these systems that they've studied is you get a shift where you'll get more arbuscular after the cutting compared to the ectos. And you can see that in clear cut, especially the first year, you get more arbuscular than the ectos. And so that's, especially if you're thinking about, well, we want Douglas fir that hangs out with ectos. You don't want a bunch of arbuscular taking over resources in the soil system. And so there's that to consider too, that you need to maintain. So in the 10%, 30%, 60%, they were maintaining enough of the trees that had the mycorrhizal, the ectos, and the other shrubs and plants that were associating with the ectos as well. So clear cut was not good for the ectomycorrhizal fungi. And so even though, when you clear cut, you definitely have more light shining on that swath of land, it just wasn't conducive for getting the Douglas fir back, bouncing back. So this was an important point they made too, that the shrubs and trees that support, that they hang out with ectomycorrhizal fungi, so they're like a resource, a reservoir for mycorrhizal fungi. They were nailed as well. In the clear cut, they got decimated. They went from 81% coverage to 6%. And so that clear cutting not only was taking the Douglas fir trees, of course, but all the shrubs and trees that serve as a reservoir for the ectomycorrhizal fungi. So that was not good. And they said, downline from that, if you reduce all of that, you're going to have reduction in fruiting bodies, reduction in spore levels. And so you're just going to be fighting to get, the ectomycorrhizals established back in that area again with the clear cutting method. So overall, their conclusions were showing that getting some of those legacy trees to stay put, not cutting everything down, and especially in the 30 and 60% plots that they tested, it did a better job of making sure that there was enough, good amounts of ectomycorrhizal fungi. There was shrubs and trees that supported the ectomycorrhizal fungi as well. And the forest floor was better protected for enhancing growth of Douglas fir in particular. So one of the quotes taken from their conclusions, it suggests that dispersed retention of overstory trees where seed trees are spaced about 10 to 20 meters apart, and aggregated retention where openings are less than 60 meters in width will result in an optimal balance of seed source proximity, inoculum potential, that's the ectomycorrhizal, and resource availability, where seedling regeneration, plant biodiversity, and carbon stocks are protected. So this is one of the first papers I've seen where they're really finally collecting good data, like solid experimental design, good solid data, and good conclusions to think about how do we do forestry a little differently and maintain the diversity in these sites, but still turn a profit. And so I was super excited to see this. And now the goal will be getting this information translated into real life, which can take a while. That often doesn't always immediately get out into the real world, unfortunately. And I don't know how, do you think foresters would be open to this idea? You're as pessimistic as I am. One thing about this, 10%, 30%, 60%, it makes your job harder, right? If you're the one out there cutting, to do that, you have to slow down and like pre-mark everything and then go in there. And and one other point she made, which I don't know, what do you think? She said it would be better if they did hand felling rather than machine felling. And again, that would be pretty challenging in today's forest industry, I think. They should bring horses in. That would be my pick. Bring the horses in. They can do a good job. So, yeah, could they adopt this? Maybe when the Douglas fir forests are almost gone, they'll adopt it. Sorry, I'm pessimistic, but I don't know. I don't know how she does it. She's got all this good data, but whether or not she'll get listened to, I don't know. But anyways, so this is the abstract. So what we just covered is pretty much said in the abstract. This is the first part of it, where she gets into how it was done, what they were collecting. And then this is the last part of the abstract. Or no. Oh, sorry, I forgot to talk about this as part of the methods. Um, yeah, I forgot to tell you this. Sorry. So it was a 116-year-old plot of Douglas fir. And but you can see that it was a mixed forest system. Western red cedar, larch, hemlock, grand fir, white pine, ponderosa pine, paper birch, and then the understory shrubs. So nice mixed forest with different canopy levels. Okay. I can post this paper, but the PowerPoints really are getting at the main points, I guess, of what I want you to grasp from this. So hopefully there'll be more work on this, but this is something where she's showing that if you can maintain a few of the older trees that can produce seeds, they're into the stage, they're mature, they're going through sexual reproduction, they're producing seeds regularly. So stick with some legacy trees, making sure that you have enough trees and shrubs that support the mycorrhizal fungi that hang out with these trees. That's the way to go. Okay. All right, so these are some of the forest studies that I wanted to talk about, and this is kind of an applied research focus on what can we do with this knowledge and how can we apply it in the real world. And so I'm also going to get into some of the other studies that have been published on plant-plant communication. A lot of the plant-plant communication really though has to do with if they're getting challenged by insects or some other pest or predator that's attacking them. So it's kind of a mix of, we have to think about plant insect or plant predator interaction too as we talk about this. But that will be the next slide set that we get into, and some of it will be field-based and some of it will be lab-based. And a lot of it will be on sagebrush. So that's a fun plant. All right, one last thing before we leave. I wanted to show a very short video. And I will post this video into EduCat. And I'll post a couple other videos. So Dr. Simard has some TED Talks that are really good, where she discusses some of this work. And so that'll be another way for you to learn the material. So I will post those into EduCat as well. All right, I'm going to do this.